Hi, welcome to today's AP Biology lesson. I'm Allison Kate, and let's get started with our warm-up today. So yesterday you were given a question for homework, and it is a visual representation of a process that is essential to gene expression. So it's showing you a few things to look at, and the question is, which of the following best explains what strand X represents? So the arrow is pointing here, strand X, and it's asking you to look at this strand, and I'm going to ask you to pay attention to the directionality. So here's the 5' end, and here's the 3' end, and notice what nucleotides are making up strand X. Okay, so again, which of the following best explains what strand X represents? Is it A, a complementary RNA sequence because it contains thymine, B, the coding strand in this process because it is being read 3' to 5', C, the antisense strand because it is serving as a template, or D, the pre-mRNA because it does not yet have a GTP cap? Okay, so hopefully you have your answer. And the correct answer is C. It is the antisense strand because it's serving as the template. And let's also look at why the other ones are incorrect. So the first choice, RNA, because it contains thymine. Hopefully you recall from the lesson that I gave you on Tuesday that RNA does not contain thymine. Okay. Um, this is not the coding strand because the coding strand is the DNA strand whose base sequence corresponds to the sequence of the RNA transcript. And it is not D, again, because it is not RNA. Strand X contains thymine. Okay, so hopefully that one didn't fool you. Okay, so today's lesson is on the regulation of gene expression and gene expression and cell specialization. And this is a difficult topic, so I'm going to try to make this as understandable as possible. We're also going to be looking at Science Practice 2C, which is about explaining how biological concepts or processes represented visually relate to larger biological principles, concepts, processes, or theories. So today we're going to look at some visual representations of the regulation of gene expression. From yesterday's lesson, you should be familiar with transcription, messenger RNA processing, and translation. And today, some important terms are going to be operon, promoter, and epigenetic. And don't worry if you're not familiar with these, I will be using them a lot today. So the objectives, by the end of this lesson, you'll be able to describe the types of interactions that regulate gene expression. You will be able to explain how the location of regulatory sequences relates to their function. That's location of regulatory sequence relates to their function. To explain how the binding of transcription factors to promoter regions affects gene expression and/or the phenotype of the organism, and explain the connection between the regulation of gene expression and phenotypic differences in cells and organisms. So here's what you need to know. First of all, let's talk about what a regulatory sequence is. And obviously we're looking at a eukaryotic piece of DNA. We're going to talk about regulation of gene expression in both prokaryotes and eukaryotes, but we're looking first here at a stretch of eukaryotic DNA, and regulatory sequences are stretches of DNA that interact with regulatory proteins to control transcription. So let's kind of dissect what we're looking at here. So first of all, each eukaryotic gene has a promoter, which is a DNA sequence where RNA polymerase binds and starts the transcription process proceeding downstream. Okay, so there's the promoter, and notice that it is labeled green. This is like green for go. This is the start of transcription. This is where RNA polymerase, which is the enzyme that facilitates the transcription of the mRNA transcript from DNA. That's where it will start. Downstream means to the right of that promoter, and upstream means to the left of that promoter. In addition, there are a number of control elements that are involved in regulating the initiation of transcription. These are DNA sequences located near or far from the promoter. The ones that are closest to the promoter are called proximal control elements, and the ones that are farther away are distal from the promoter, and distal control elements can be grouped together, and they are called enhancers. Okay, so you can see that there are several distal control elements grouped together as an enhancer, and that's farther away or farther upstream from the promoter. And then we've got our proximal control elements that are proximal to the control to the transcription site. Did I say distal? Okay, so distal is farther away and proximal is closer. Okay, so at the other end of the gene, there is a poly A, which stands for many adenine signal sequence in the last exon of the gene that is transcribed into an RNA sequence. And this is the signal where the transcript is cleaved off and the poly A tail is added. I'll talk about what that poly A tail is in a minute. So look at the poly A sequence. It is in that last exon, and that's the signal for where we are going to stop that transcript. All right, so one other thing that's going to happen is once we get our primary transcript, it gets processed, and it will get processed in 3 steps. The first step will add a guanine cap at the 5' end of that mRNA. The next thing that will happen is there'll be an addition of a polyadenine tail at the 3' end. And both of these things are going to protect this messenger RNA transcript once it leaves the nucleus. And then there's some splicing that goes on. The introns, which are the non-coding segments, are going to be removed, and the exons will be connected together. Oops. Sorry. And so I also wanted to point out, so we remove our introns, we connect our coding segment, which are the exons, and also here you're going to see this UTR, that's an untranslated region. And so we know that we're going to, when we finally get out to the ribosome, we're going to start translating this mRNA here and end it here. So there's also epigenetic changes that can affect gene expression through reversible modifications of either DNA or histones. So 2 things to unpack in that sentence. First of all, epigenetic means above the genes, literally. So this is not involving a DNA sequence that is going to be changed. So there is some other way of regulating gene expression. And so again, we're in eukaryotes, right? And in eukaryotic DNA, the chromatin is packed using histone proteins. And so what we're going to do is we're going to see that there are some ways to either loosen or tighten the chromatin to either have greater access to those genes or less easy access to those genes. So the inheritance of traits transmitted by mechanisms that are not directly involving the nucleotide sequence is called epigenetic inheritance. And so if you look at the image, amino acids in the histone protein tails may be chemically modified. And I'm illustrating the acetylated histones. So acetyl groups are represented by these green balls, and they may be added to the chromatin to be less compact, which makes the DNA more accessible for transcription. And so you can see where those acetyl groups are, the DNA is less tightly packed together, which you can see in this unacetylated histone. The phenotype of a cell or organism is determined by a combination of genes that are expressed and the levels at which they're expressed. So I'm going to give you an example. And what you're going to see in this example is I've got 2 different types of cells, a liver cell and a lens and eye lens cell. And we can see that on the actual piece of DNA, on the gene, we have both the albumin gene and the crystalline gene. However, the albumin gene will only be expressed in liver cells. It is a blood protein, and the lens cells will only produce the crystalline protein that's for the eye lens. And the way that that happens is in each cell there are different specific transcription factors that the cell make that will determine which genes are expressed. And so if you look at my arrow here, I'm showing you that in this picture we've got just different colors representing the different transcription factors that are able to activate the transcription of this particular albumin gene. And so just to color code it, we've got a red, a gray, and a yellow. And they are going to actually, and I'm going to talk about this bending that's going to happen in a future slide, but they are going to actually be able to activate, this is RNA polymerase, to activate it to begin to express that transcribe that albumin gene. And that does not happen in the crystalline gene, even though those genes are both present in the liver cell. In the lens cell, there are different activators that are available, and these are color-coded pink, gray, and brown, and you can see that they're going to get activated. That's going to allow the RNA polymerase to transcribe the crystalline gene, but not the albumin gene. So that explains why you get different proteins expressed in different types of cells, even though the genes are present in both. Okay, so both prokaryotes and eukaryotes have groups of genes that are coordinately regulated. We're going to talk about prokaryotes first, and then I'll talk about eukaryotes. So in prokaryotes, groups of genes called operons are transcribed in a single messenger RNA molecule. So an operon is a group of functionally related genes that can be coordinately controlled by a single on-off switch, which is called the regulatory switch. This is a segment of DNA called the operator, which is usually positioned within the promoter. And I'm going to give you a graphic to illustrate this. So an operon is the entire stretch of DNA. That includes the operator, the promoter, and the genes that they control. There are two types of operons that I'm going to talk about today in prokaryotes. Repressible, which is a system where the gene is on, but you can turn it off or repress it. And the tryptophan or TRP operon is an example of that. Or an inducible operon, which is a system where the gene is normally turned off. It's not being expressed, but it can be induced to turn on. And the lactose or Lac Operon is an example of an inducible operon. So let's take a look at both of these. First, we're going to take a look at the Lac Operon, which is inducible. Again, it means that the system is not transcribing the gene needed to break down a sugar, lactose, which is a disaccharide, but it can be induced to do so. So first, let's look at when lactose is absent. The repressor is active, which means that it is repressing the transcription of the genes, okay? So the operon is off. And if you look at what's going on here, we've got our DNA. This is the promoter, the operator, and then we're going to have our structural genes here. This is the RNA polymerase that is responsible for transcribing. So when lactose is absent, there is no need to break it down. So you don't need to make those proteins or enzymes that are going to break it down. And so the RNA polymerase is blocked by this repressor, which is sitting in this operator. In the second case, okay, so in the absence of lactose, the repressor switches off the operon by binding to that operator. In the second case, lactose is present. And when lactose is present, it is a nutrient for that bacteria, so it needs to break it down to digest it to get its energy. In this case, we're going to look at an isomer of lactose called allolactose, and it's being represented by this green ball here. And it is going to act as an inducer. It's going to bind to the repressor protein, so you can see that in this part. So it's going to hop right in this binding site, and it changes the shape of that repressor. When that shape is changed, it can no longer fit in the operator, and so the block for the enzyme RNA polymerase is removed. So RNA polymerase will hop on the gene, and it will begin transcription, and it will transcribe these three genes, LAC-Z, LAC-Y, and LAC-A, making the enzymes that are necessary to digest lactose. Okay, and that makes sense because when there's no lactose around, you don't need to make those genes to digest it, but when lactose is present in the bacteria's environment, then they need to turn that system on. All right, let's look at The repressible type, which is represented by the Trp Operon. Tryptophan is an amino acid, so it's similar. It is a necessary nutrient for the bacteria. Again, repressible means that genes needed to synthesize tryptophan are being transcribed, but the system can be repressed. Okay, so in this case, we still have our piece of DNA. We've got our promoter and our operator. And this time we have 5 genes that are going to make the polypeptide subunits that make up enzymes that are needed to make tryptophan. Okay, so when tryptophan is absent, the operon is on. That means that there's no tryptophan in the bacteria's environment, so it needs to make its own tryptophan. Okay. And the reason that that works is that here is the repressor. It is not bound to the operator, so that operon is on. Okay, now when tryptophan is present in the environment, we no longer need to make it on our own. So we can turn this system off. And so what happens is tryptophan itself is the co-repressor. And if you look, it's a conformation thing. Here's tryptophan, the co-repressor is going to fit into the repressor, and it is going to change its shape. Okay, so now its shape is compatible to fit into the operator, and now when RNA polymerase tries to transcribe this gene, it can't fit on the operator or the promoter, so it's blocked. So that means that no tryptophan will be made, and that makes sense. Since tryptophan is present, the organism does not have to waste any resources making it. Okay, so now we're going to talk about eukaryotes. In eukaryotes, groups of genes may be influenced by the same transcription factors to coordinately regulate expression. Since operons are not used in eukaryotes, the genes that are co-expressed are scattered across different chromosomes. So for coordinated gene expression to occur, it depends on every gene in that dispersed group having a specific combination of control elements. It often occurs in response to a chemical signal from outside the cell. It could either be a steroid hormone or a protein hormone that activate transcription factors. And as you might remember from your lesson on cell signaling, steroid hormones act directly to activate or repress transcription factors, whereas protein hormones are going to act via a signal transduction pathway to either activate or repress those transcription factors. So let's take a look at a few examples. So promoters are DNA sequences, again, upstream from the transcription start site where RNA polymerase and transcription factors bind to begin or initiate transcription. So now let's take a look at our DNA. Again, we've got our distal control element, which is farther away from that promoter. And these are our enhancers, and we have our activators, right? We also have our promoter, and this is the gene that we want to transcribe, okay? So what's going to end up happening is to regulate the initiation of transcription, the DNA control elements in the enhancers are going to bind to these specific transcription factors. And that's going to facilitate the bending of this DNA, and that's going to enable those activators, okay, right here, those activators to actually contact the proteins of the promoter and will initiate transcription. So once the activators have attached to those mediator proteins, then we have a transcription initiation complex. And as you can see, RNA polymerase will be able to bind to the DNA, and it will begin transcribing that gene. So there are some negative regulatory molecules that inhibit gene expression by binding to DNA and blocking transcription. And the main way that I'm going to talk about here is through DNA methylation, which greatly reduces transcription because it's going to prevent the RNA polymerase from actually getting into that gene. So what happens in DNA methylation is we're going to add some methyl groups to certain bases in DNA, and it's usually cytosine. So if you look at my image, you can see that there are methyl groups that are attached to those cytosine nucleotides. And what will happen is in this actual methylated piece of DNA, it actually restricts RNA polymerase from binding, okay? And then without that methylation, you can see that RNA polymerase has more access to the DNA. So individual genes are usually more heavily methylated in cells where they're not expressed, right? That makes sense because they're harder to get to. Once methylated, genes usually remain so throughout successful cell divisions. And so after replication, enzymes methylate the correct daughter strand so that the methylation pattern is inherited. So gene regulation results in different gene expression and influences different cell products. So let's take a look at, this is a gene for a muscle protein called troponin T. And we can see that it has 5 exons. So, right, these light blue are going to be introns and removed. So we're going to end up, after we do our mRNA processing, we are going to use these exons to put together or splice, and we can put them together in different patterns. And this is called alternative RNA splicing. So if you notice in the first RNA, this is the transcript that we get after this DNA is transcribed. And the first one, mRNA molecule has ended up with exon 3, which is shown in green, and the other one has exon 4, which is shown in purple. And you can see that these two mRNAs are translated into 2 different but related muscle proteins. So in this way, we can actually transcribe it once and then splice it 2 different ways to get different proteins produced. There are certain small RNA molecules that have roles in regulating gene expression. These are miRNA, which stands for a small single-stranded RNA molecule called a microRNA. Okay, so we're showing it here in yellow, and this is going to be our transcribed mRNA. And we're going to actually, those miRNAs actually degrade the messenger RNA. And so if the mRNA is degraded by these microRNAs, the match is not as complete when so you can see here, if we're looking at the hydrogen bonds that are holding these together, translation at the ribosome will be blocked. So the miRNA has a role in breaking down the mRNA to be translated. So a review. There are lots and lots of ways to regulate gene expression in both prokaryotes and eukaryotes. In prokaryotes, operons are used. In eukaryotes, we see a variety of methods. The first one is through chromatin modification. Okay, so normally in eukaryotic DNA, the genes are wound up around these histone proteins, and so we can either loosen or tighten that to give access through histone acetylation, right? That loosens up the chromosome structure to enhance transcription, or through DNA methylation, we are going to add those methyl groups, which blocks the ability of RNA polymerase to transcribe the gene. So that reduces transcription. Then we can also look at what goes on in transcription. Remember to regulate the initiation of transcription. There's DNA control elements, the enhancers and activators facilitate the bending of that DNA to connect the activators to the proteins at the promoter, and that will initiate transcription. Okay, genes that are in a coordinately controlled group all share a combination of control elements. Then we also know that there's both mRNA degradation, which I just described through the process of those microRNAs, There's also protein degradation and processing that occurs after translation, and that can be post-translational modifications. And then finally, in the middle here, we have RNA processing, which you probably heard about yesterday. But again, you can once you process your primary transcript, you can also do alternative RNA splicing, where you are able to transcribe one gene, but then splice those exons together in different ways. So let's review a whole signal. So let's say this is a signal, maybe it's a steroid hormone that is going to signal the activation of some transcription factors. So the signal arrives at the nucleus where the DNA is going to, where it activates some transcription factors, and we are going to then transcribe the gene. We get our primary transcript, which is then processed. We get that cap and tail, which protect it as it leaves the nucleus for the cytoplasm. And then maybe those microRNAs degrade the mRNA, and so it is not transcribed. Or if it's transcribed, we get our protein. We get an active protein that may, in fact, be transported outside of the cell or used within the cell, or that protein could be broken down. So there are many places along this entire pathway where gene expression can be regulated. All right, so let's put this into some practice. So first of all, let's take a look at what happens in prokaryotes. And in this, bear with me because this is kind of a long question, but be looking at the shapes. Remember, our skill is to look at some models that are represented visually. Okay, and so what you're looking at is the Lac Operon in E. coli. It consists of genes that code for enzymes necessary for the breakdown of lactose. When lactose is absent, the operon is inactive because a repressor protein binds to a specific site on the Lac Operon. When lactose is present, lactose molecules bind to the repressor protein, causing the repressor protein to dissociate from the binding site. In the absence of glucose, which is the preferred energy source for bacteria, the protein CAP or CAP binds to a regulatory site near the Lac promoter to activate transcription of the Lac Operon. The following symbols represent macromolecules involved in the regulation of the Lac Operon. So you can see we've got a repressor that is a circle. We've got two 6 carbon rings representing the disaccharide lactose. We've got a triangle for CAP. We've got an oval for RNA polymerase, and then we have a squiggly line for the messenger RNA. In the diagrams below, the horizontal line represents the Lac Operon, and the bent arrow represents the transcription start site of the Lac Operon. Here's your question. Which of the following diagrams best represents the scenario in which lactose is available to the cell and glucose is absent? So I want you to think about that. Glucose is absent, lactose is present. Which of those diagrams, A, B, C, D, best represents the scenario? Okay. So the correct answer is C, and that's because what you see is the lactose is actually bound to the repressor. So there is no repressor in the way of RNA polymerase, and it is actually making that messenger RNA. So it's the only one where you see the messenger, the transcription, the product of that transcription. What's wrong with D is it's showing that the lactose is actually bound to RNA polymerase, and we know that that is not how the system works. Now let's take a look at a practice question in eukaryotes. We again practicing our visual representations. The figure above depicts the DNA protein complex that is assembled at the transcriptional start site of gene X when the expression of gene X is activated in liver cells. Previous studies have shown that gene X is never expressed in nerve cells. Based on the diagram, which of the following most likely contributes to the specific expression pattern of gene X? Okay. So we've got A, expression of gene X produces large amounts of transfer RNA, but undetectable amounts of mRNA. Think about yesterday's lesson. B, the general transcription factors inhibit the activation of gene X in liver cells by blocking the activator from binding to RNA polymerase II. C, the activator is a sequence-specific DNA binding protein that is present in some tissues but not others. And D, the enhancer is a unique DNA segment that is added to the nuclear DNA of some cells of an organism during the process of mitotic cell division, but not others. So if you're paying close attention to today's lesson, you should have your answer. And of course, that answer is C. And that is because we are seeing that the activator is a sequence-specific DNA binding protein that is present in liver cells, but not nerve cells. Okay. All right, so now let's get a FRQ practice. A difference between prokaryotes and eukaryotes is seen in the organization of their genetic material. Contrast the following activity in prokaryotes and eukaryotes. And what you are going to do here is contrast how genes are regulated in both prokaryotes and eukaryotes. Okay, so hopefully you have some ideas about how to approach this question. So let's take a look. Okay, so in prokaryotes, we use operons, right? So we have one single on-off switch in a sequence in DNA that's all together. In eukaryotes, there is no operon. In prokaryotes, we have both positive and negative control. In other words, we can induce or repress those genes from being transcribed. In eukaryotes, if you think about it, our control is primarily positive control. In other words, the gene is not transcribed until that transcription initiation complex is in place. In prokaryotes, enhancers are rare, and as you saw, in eukaryotes, enhancers are commonly used. In prokaryotes, because there is no tightly packed with histone DNA, there is no other type of epigenetic regulation or above the gene sequence regulation. There's methylation, which you might remember will make it more difficult to transcribe genes, and there's acetylation, which will make it a little bit, will make the DNA more accessible to be transcribed. Okay, so that's kind of a good slide to think about and remember. All right, so let's do a check for understanding, specifically for Science Skill 2C. Again, we're looking at a visually represented process, right? We're looking at gene regulation represented visually, and your task is to be able to explain how biological concepts work in this visual representation. So you're looking again at that Lac Operon, and we are looking at lactose being utilized for energy by the E. coli when glucose is not present. Allolactose is an isomer of lactose that's in the environment of these bacteria when lactose is present. And then, of course, the cap site prevents the binding of RNA polymerase when glucose is present in the environment. So then we see we have the LAC-Z, Y, and A genes, which code for the proteins that are needed for lactose metabolism. So what you are going to do is look at this. And again, if you look at allolactose, here's the repressor, here's the operator. We can see that RNA polymerase is here, and we can see an arrow. Here we see allolactose absent. And so the repressor is in the operator. Repressor is not in the operator here. And we can see that RNA polymerase is not transcribing. We've got a big X there. So this is the question. Which is a scientific claim that is consistent with this information that is presented visually in figure 1? And I'll come back to this, but let's look at our answer choices. So first of all, the presence of excess lactose blocks the functioning of RNA polymerase in this operon. B, when bound to the operator, the repressor protein prevents lactose metabolism in E. coli. C, the binding of the repressor protein to the operator enables E. coli to metabolize lactose. And D, allolactose acts as an inducer that binds to the operator, allowing E. coli to metabolize lactose. Okay, so here's the correct answer, which is B. And it's when bound to the operator, the repressor protein prevents lactose metabolism in E. coli. Okay, so let's go back. When bound to the operator, okay, the repressor prevents the RNA polymerase from transcribing that gene. Okay? If the gene can't be transcribed, it cannot make the enzymes necessary to break down lactose. All right, so here's your debrief and summary. Today, you now should be able to describe the types of interactions that regulate gene expression, explain how the location of regulatory sequences relates to the function, explain how the binding of transcription factors to promoter regions affects gene expression and/or the phenotype of the organism, explain the connection between the regulation of gene expression and the phenotypic differences in cells and organisms. And our key takeaway today is that differences in the expression of genes account for some of the phenotypic differences seen between organisms. So let's talk about some common errors and misunderstandings about gene expression and regulation of gene expression and cell specialization. This was an FRQ that was asked on the last year's 2019 AP Bio exam. And so what we're looking at is a visual representation of some data. So a researcher is studying the patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from 6 genes. And the data are shown in this figure. First of all, you need to look at the legend. The open squares, no mRNA is seen. The gray squares, a moderate amount of mRNA is seen. And in the completely blacked out squares, a high amount of mRNA is seen. So across the top, you can see we have 6 genes called E, F, G, H, I, and J. And then we have tissues, liver, heart, brain, kidney, pancreas, and skeletal muscle. So you need to look at this data. And then in part A, you are asked to identify the gene that is most likely to encode a protein that is an essential component of glycolysis, and then provide reasoning to support why you selected that gene. And then in part B, the researcher observed that tissues with a high level of gene H mRNA did not always have a gene H protein. Provide reasoning to explain how tissues with a high gene H mRNA level can have no gene H protein. So there are 2 different things. First of all, you need to identify the correct gene that is most likely going to encode a protein essential for glycolysis, and then figure out why, if you have the mRNA, you don't have the protein for gene H. Okay, so let's look at the scoring guidelines. This was a 3-point question. The first point was identification, and you simply needed to identify gene G. Okay, and the reasoning behind that was also worth 1 point, and you, for either one of those bullet points, you would be awarded a point, and that is that gene G is the only gene expressed in all 6 tissues, and a glycolysis occurs in all 6 tissues. So you needed to make that connection. Just saying that it was expressed in all 6 tissues was not enough. You needed to connect it to the fact that all 6 tissues undergo glycolysis. Or you could have said that mRNA is the only mRNA present in all 6 tissues, and glycolysis occurs in all 6 tissues. Okay, and then finally, for your third point, you needed to provide reasoning for why you have the mRNA from gene H, but you don't have the gene H protein. And any one of those bullet points would have earned you a point. The mRNA is not exported from the nucleus. The gene H mRNA is not translated, or those microRNAs could have prevented translation, or there could have been other post-transcriptional modifications to the mRNA that was transcribed, preventing that protein from being produced. Okay, so let's look at let's look at a student response that earned all 3 points, and then we'll look at where the knowledge gaps and misunderstandings were. So here's a 3-point question. 3-point answer, excuse me. Gene G is most likely to encode a protein that is an essential component of glycolysis because the mRNA is at least moderately present in all types of tissues. All tissues undergo glycolysis to get a small amount of ATP to function. Gene G is the only gene that has mRNA present in all tissues, so all of these tissues are able to code for the specific protein. Okay? And then for part B, tissues could have a high level of gene H mRNA, but not have a gene H protein because the mRNA was never translated. The gene H may have been transcribed from DNA to mRNA, but if the rRNA and tRNA do not translate this particular strand, it will not code for the amino acid and therefore not become a functional protein. So excellent 3-point response. So where were the knowledge gaps? A lot of students had a difficult time correctly identifying gene G, and also not understanding that gene G is the only one that was expressed in all 6 tissues and that glycolysis occurs in all 6 tissues. So making that connection was crucial. So a response that demonstrates understanding of that is that gene G has a high to moderate amount of mRNA in every tissue on the table. Because glycolysis is the first step of cellular respiration, which is ATP making, gene G would most likely encode a protein essential for this energy-bringing process, which is done throughout the body. So making that connection between gene expression and its function. And then in part B, students had difficulty correctly reasoning that gene H mRNA is not exported from the nucleus, or it's not translated, or that there's some type of RNA interference preventing translation, or that it undergoes those post-transcriptional modifications. So this was a difficult part of this question for students. Here's an example of a response that demonstrates understanding. Tissues with high gene H mRNA levels can have no gene H protein, as unsuccessful translation can lead to the lack of gene H protein production. Okay, so I'm going to give you your homework, but before I let you go, I'd like to give you just a few tips for getting ready for your AP exam on May 18th. So here's your homework, and this is due tomorrow. So Ms. Pfizer will be with you tomorrow, and she will go over the scoring guidelines for this. Human cells divide every day for normal growth, replacement of cells, and tissue repair. And you know that if you joined our lesson on cell cycle and mitosis. The dividing cells proceed through 3 phases of the cell cycle as shown in the figure. You see mitosis, cytokinesis, and interphase. When a person experiences a cut, damaged skin cells secrete a protein called epidermal growth factor, EGF. Epidermal, which are skin cells in the vicinity of injury, possess these proteins in their cell membranes called epidermal growth factor receptors, or EGFR. Okay, so there's your background. You can read that as many times as you need to understand kind of the background to help you with the intention of this question. Cancer can result from disruptions in the cell cycle control. Mutations that increase the production of EGFR have been associated with skin cancer of the head and neck. Describe, that's your task verb, how a mutation outside of the coding region of a gene that codes for EGFR could lead to the overproduction of the EGFR protein in a skin cell. So this is going to have you put together some lessons on cell cycle regulation and control, and also think about the expression and regulation of eukaryotic genes. Okay? So again, that will be your warm-up tomorrow. So just a couple of slides here, just so you can kind of get a visual of what you're going to be seeing in the next couple of weeks. So you will need an AP Biology exam e-ticket. There are 2 ways that you'll get this. You'll either get this emailed to you 2 days before each test. This one happens to be for biology, but if you're taking multiple AP exams, you will get your test ticket 2 days before your exam. If you don't get the email, this is why we've been bugging you to go and make sure that College Board has your most current email on your AP account at MyAP. You can go to MyAP at collegeboard.org and make sure that your email is updated. Okay. Otherwise, you'll be able to get it at your My AP at the College Board. Just a reminder about knowing when your test will be administered. Okay, know your time zone. AP Biology exam will be given on Monday, May 18th at 2 p.m. Eastern Daylight Time. So I'm in California. My students will be sitting for the exam at 11 a.m. If you are in Madrid or in a European time zone, check your time zone. You may be taking that test at night, so make sure you take a nap during the day. Be prepared. Make sure you understand your time zone. If you miss, then you will have to figure out how to get into that makeup exam. All right, and then 5 quick steps to prepare for your exam day. Again, review your contact information, check your tech. We've been asking you to make sure you have a device and internet connectivity. We are, and I'm going to talk to you more about this next week, beginning on Monday, May 4th, there will be an exam demo. It will not just be for AP Bio, it'll be for all generic AP, and you can practice how to submit your response. So you're going to be walked through how to do that and the 3 different ways that you're going to be able to write your response. Okay, and more on that later. Make sure that you gather what you need for each exam. You can consult collegeboardcb.org/exam day docs, download them, print them, gather your additional materials for your specific exams. For example, for AP Biology, you might want to have that AP Biology formula and equation sheet handy. And then make sure you receive your exam confirmation and e-ticket emails. Okay? So, final words. During this challenging time, the AP program wants every interested AP student to have the opportunity to earn college credit you've been working toward all year. So please stay safe and healthy, and remember, you got this. All right, so thanks for watching. Look at your checklist, complete your checklist. We hope you'll join us again tomorrow for the last review lesson. That'll be with Ms. Pfizer. She'll go over your practice question. And tomorrow's lesson will review mutations in biotechnology. And I'd like to wish all of you best of luck on Monday, May 18th. Thanks for watching.